the microbial growth. So the lag, exponential, stationary phase, and death phase, everything I have told about. But you should know exactly how the growth of your microorganism will occur. So you consider that we will make in our laboratory, we are going to use some of the nutrient broth, and then we have inoculated the culture a day before. Then after a day before, we will get a subculture, okay? Because everything will be all the microorganism plate, the agar plate or culture, everything will be stored under minus -3 and all. So whenever we required a fresh culture, we should prepare a day before the subculturing of your organism. So you will prepare if you have lab tomorrow, then we have to prepare every samples today itself. So we will exactly take, you know about the inoculum preparation, right? So you have to exactly take the single colony and you should inoculate into your nutrient broth. So that nutrient broth will send some of the nutrients to your organism. If you kept it for the overnight, then the subculturing method, inoculum method will be achieved in incubation of 37 degrees centigrade in a shaker flask. Okay. So from that, the day one, that is the exact where we have a lab, then day one we what we will do is that same, like you have to autoclave your conical flask, you have to prepare a fresh medium, then you will inoculate some amount of your sample, that is subcultured sample, into your conical flask. Okay, then you have to mix it out and you have to take a initial OD. If you took an initial OD by using a UV spectro or a calorimeter, you will get only the 0.00 some values of sometimes zero itself. So based upon the culture you have added, then you will keep it for the shaker for every half an hour to 1 hour. When you are checking the values of your OD, there will be a curve when you plot it against the time as well as the number of bacteria present. You will get a curve-like structure which will represent the growth of your microorganism. So, in this graphic, they have shown different stages. That is lag, exponential, which is also called as log phase, stationary phase, and death phase. If all the nutrients have been depleted from your medium, then the death phase will occur. So what is lag phase? Whenever we are using or else whenever you are inoculating your organism into a new environment, it requires to adapt to that particular environment. It's same like our case. Like if we are getting into a new place or if you are moving on to your hostel new rooms and all, we required some time to adopt ourselves into that room and to keep or else to adapt to the people who is present in that room. It's same like the microorganism will start adapting to that environment, which is very very rich in nutrient, and it will increase its size. It will start utilizing the nutrients from the medium, and it will increase in the size. But there is no any solution occurs. So this is called lag phase. Okay, during that time, the growth and the cell division has been affected, so we will get only the minimum line of, that is in 1 hour we will get only the not drastic change in our OD value. Then exponential phase. So in exponential phase, once it have been adapted to the environment, it will start dividing itself. So doubling of microorganism will occur in exponential phase. So while the doubling occur and also the existing microorganism will undergo utilizing of the nutrients, and its metabolic activities will also occurs inside the solution. So during that metabolic activities, and also if we have added the antibiotics into your media, and also that will effectively prevent that organism to be very much specific to your organism. There is no any cross contamination occurs, and resistant gene will also be on cell division. It will also have some of the resistant gene. As we all know, we have studied in the cell biology itself what is exactly the cell will do. So the cellular process, the cellular thing, all the organelles present in your cell will start what we can say. It will have each and every functions. We remember in already in cell biology itself. It will have each and every specific function. That specific functions, metabolic functions, cellular functions will be carried out by the microorganism on seeing on utilizing the nutrients. Okay, so this is what happened in the log phase. So all cell division, utilization of nutrients, metabolic activities, then and also the primary metabolites it will start producing to enhance its own growth, to enhance its own growth. Okay, stationary phase. In stationary phase, we can say after the exponential phase, the microorganism that is doubling of microorganism and also the growth of the microorganism, utilization of the nutrients will start minimizing, because almost the microorganism present in the medium will start utilizing the nutrients in the medium, so there may be a depletion of nutrients occurs for that purpose. And also the microorganism, because of its metabolic activities, the accumulation of the waste product will also occur inside your media, because it is completely a batch process. Once you have once you have prepared the media, you have inoculated it, you will close down everything and you will keep it as such. You're not going to add any of the nutrients in between. So because of that, in stationary phase, what will happen, the number of cell division will be, and also the number of cells died will be equivalent. So they will have a steady phase off. There will be cell division, and also the cell which have been produced before, they will lack of the nutrients, it will start dying. So both the division as well as the dying will be almost equal. Number of cells dividing and death is almost equal. So the overall population will be maintained stable, very steady state. And even it will also produce some of the secondary metabolites. So that will also occur in the stationary phase. And this is what in stationary phase, if the microorganism will have been used for the spore forming bacteria and all, this phase will generate some of the that virulent factor we can say the secondary metabolites. That secondary metabolites can able to also it will act as an very bad environment for the organism which is present inside your stationary phases. The possibility of dying may be because of two things. One is depletion, another one is the secondary metabolites, excess amount of secondary metabolites, which may also create a very bad environment for the same organism, so that it may die. So this is what the stationary is. So after utilizing everything, there is no initial division occurs. The accumulation of product, our desired product, or all the glucose or nutrients present in that is converted into a desired product, and also the excess gas and also byproducts. Because of the accumulation of all these things, the microorganism will start dying. There is no any excess nutrients present in the medium, so it will start dying. It once it is dying, then obviously we know that the cell wall will be get break and all the organelles will come throughout your samples. So this is what the exact the growth phase is. Okay, from the graph you will know about. So in lag, it have been adapted to it. In log phase, it will start multiplying and dividing. The growth of the organism will be there along with the metabolic activities. In stationary phase, the division as well as the death will be almost similar, maybe because of the new depletion in the nutrients and also the metabolic activities. Then there is no any nutrients present in that medium, so organism will start dying in the death phase. This is about the growth.